Good afternoon, everyone. I'm going to talk to you about, again, the theme about collaboration. I'm going to talk about collaborative framework that I utilize to advance my program of research in fatigue. So it all started, my trajectory started in my dissertation, which is understanding the quality of life of patients with involvement of their sarcoidosis. We call this in Hampton as the eyeball study. That's why you see it. So along my presentations are images that can link on how I developed this collaborative framework. So this dissertation really made me appreciate the impact of fatigue, the symptom of fatigue in the quality of lives of individuals, especially those with chronic illnesses. So when I joined NINR and Dr. Mary Kerr asked me, what do you want to do? I said, I'd really love to develop a program of research in fatigue. So in order to describe a trajectory and causes of fatigue in different conditions, using clinical and preclinical approaches, and also understand what are the symptoms that cluster with the symptom experience to enable to explore and fully understand the distinct and shared pathways that can explain the biologic underpinnings of fatigue. So when I started, I had this framework in mind. I first developed a matrix of mentors, and we discussed what would be the best way, or what's the best population at first, to really look at the trajectory of fatigue. And so we came to a decision that the radiation group, the prostate men who develop, who don't usually have fatigue to start with and develop worsened fatigue, and some of them, most of them, have resolution of their fatigue after completion of treatment, is the best population to look at. And so with that, since I don't have an oncology background, I reached out to a lot of cancer-related fatigue experts in the community. Most of them are nurses. Chris Miaskowski was kind enough from UCSF to help me put together my protocol. Of course, Barbara Piper, and of course, Dave Sella from Northwestern, who developed the PROMISE to help me put together the study outcomes that I'll incorporate in my study. And of course, as the initial set of collaborators, which are NCI, radiation oncology, and rehab medicine, who have helped me with objective measures that I incorporated. And of course, the backbone of these are the hardworking fellows, the trainees in NNR, the postdoc, postbac fellows, and my special volunteers. So when I started with a natural history study, following patients during their radiation therapy and post therapy, measuring their symptoms, obtaining blood for molecular correlates. And of course, the rehab medicine helped me to put together some of the objective measures that it can incorporate to see any correlations with this fatigue experience. Radiation Oncology Department of NCI has been kind enough to open their doors and welcome me to see their patients and follow the fatigue symptoms of these patients. So over time, as I accrued patients, I was able to have initial data and really saw that there's about 37% of patients, the lower the scores, the worse the symptoms of fatigue. 37% of patients develop chronic fatigue 1 year after after completing the radiation therapy. And interestingly, though, this chronic fatigue experience is associated with their depressive symptoms, global pain, and urinary symptoms. But it's not related to their anemia. Their anemia is only related during their treatment. But during that chronic fatigue experience, it's not related to their anemia, it's not related to their body weight, nor is it related to their age. So looking at another aim is, of course, molecular correlates. We have a great space in our NINR lab, and I hope I'm sure most of you have visited us, but if you've not, please you're welcome to come by and visit us to tour the lab. So we did a whole blood microarray experiment, and we were able to identify differentially expressed genes that were associated with this chronic fatigue population. Other independent experiment was the 48 cytokine panel, which I collaborated with the National Institute of Arthritis and Musculoskeletal Diseases, a translational immunology lab. We were able to gain access into that lab, and we were able to run our experiments. And through that, we were able to see that trail, which is one of the TNF family cytokine that belongs to the TNF superfamily, was significantly increased in our chronic fatigue population. And interestingly, though, also, the trail decoy receptor was upregulated in our micro-experiments. So in order to further understand this, I have to dig deeper and see who can really help me move this initial finding forward. And of course, looking around, I was able, it directed me to Dr. Jan O'Shea. You know, Dr. Jan O'Shea is part of an Affordable Rock and Roll Act, or the ARA, with Dr. Collins. So at that time, Dr. Jan O'Shea was really interested in doing the talk about rock and roll. And they were also looking for members, because most of the members there are fellows that have gone NA, so they're looking for members, and also a front act for their concerts. And of course, my fellows who are here, who are presenting also the posters, have their band as well. They're called the Neurofarm. So I volunteered them. Caution, there's no coercion. But I volunteered them. So they're actually doing a front act for the ARA rock and roll, and they're actually talking about merging them to become the Cytokine Rock. So, yeah. But anyway, with the love of rock and roll, but more importantly for our love of science, Dr. Jan O'Shea was really helpful in opening his resources for us to really dig deeper into our initial findings. We, as you've seen probably in one of the posters, we were really excited, and we are excited about these recent findings that the glutamatergic system, especially the mGLOR5 receptor, has a major role in the association between fatigue and inflammation. And this association between fatigue and inflammation has been proposed for a number of years. But interestingly, this is the first time to see the link that glutamate receptor may have a role in the association between fatigue and inflammation. And that became really visible. The evidence is through the microscopy lab, the core lab of NIAMS, where we found really the mGLOR that clustered together after radiation, and that clustering or the activation of this mGLOR5 gave way to the increased release or concentration of a pro-inflammatory cytokine. More of that, with the NIAM support, they also gave way to the more collaboration with NHLBI. NHLBI has a great animal program, especially their phenotyping core. So to really fully understand our fatigue experience, especially from our clinical results, we really wanted to understand the phenotype. What is this fatigue behavior really involves? And looking at how is the central and the peripheral triggers play into this fatigue experience. So we were able to develop our radiation-induced fatigue model with the help of NIAMS, but also especially with the help of NHLBI phenotyping core. So our initial data are from a small number of samples here in the clinical center. We really wanted to reach out to our extramural partners to validate this in a community setting. So we were able to reach out to our friends in Georgetown Radiation Medicine Department, and they were kind enough to administer the same questionnaires as we did, and also share the samples, especially the plasma samples that they've collected. And indeed, with that, we still saw the significant association between fatigue and trail concentration in the plasma 2 years after completing a radiation therapy for the same population. Further than that, we also looked at, because these are 1 year after, we wanted to look at whether there are also significant genes that can predict patients at risk to develop this chronic fatigue 1 to 2 years after receiving or completing radiation therapy. And with that, we were able to reach out to bioinformaticians in Harvard Cybernetics Lab, as well as a thermodynamics group from Spain, to help us identify and develop an algorithm, predictive algorithm, that can help us identify specific genes that can predict patients at risk to develop chronic fatigue 1 year after completing. And indeed, that gave us more of confirmation that indeed glutamate receptors may play a role in the chronic fatigue development in this population. So with that, since the glutamate receptor seems to be a common theme that's a repeating theme in all these findings, we were able to reach out to experts within the intramural community, especially in the National Institute of Mental Health, who have used the glutamate receptor as a target for their as an antidepressant agent. So with our help, we've collaborated a lot. We developed a new fatigue questionnaire together, and I did secondary analysis of all patients that have gone through their clinical trial. And we saw from that secondary analysis that indeed, ketamine, which is a metabotropic glutamate receptor itself inhibitor, has an anti-fatigue effects. And with that, we've used this as a pilot or a proof of concept study to see whether ketamine has also an anti-fatigue effects in our population. So my model, the conceptual framework that I've developed, has grown, has expanded. I still have this strong tenured NIH investigator who helps me navigate and retain the focus. But my intramural collaboration has expanded to several other investigators across the different institutes here in campus, as well as my extramural collaborators. Most of them are trainees that graduated from our fellowship here, but also those that have graduated from our GPP programs. We've collaborated a lot related to data collection, sharing of data, but also in terms of analyzing and interpreting those data that we've collected. And of course, other senior scientists and clinicians that have reached out and are also interested in this area of science. And so these are I acknowledge the hard work of my staff in the NINR lab, as well as my intramural and extramural collaborators, but more importantly, the patients and their families for taking us in and be involved in this experience. So thank you very much.
Good afternoon. I hope you're all doing well. I'll be talking about the intramural NIH collaboration on the Myalgic Encephalomyelitis Chronic Fatigue Syndrome project. In October of 2015, Francis Collins, the director of the NIH, declared that there would be a new NIH effort to study the chronic fatigue syndrome. As he put it, medicine desperately needed some new ideas in the fight against the syndrome and the closely related neurological disorder, myalgic encephalomyelitis. At this moment, there is no test, cause, or treatment for the condition, which causes overwhelming and often disabling fatigue in more than a quarter of the people with the disorder. There is something going on, he said, and we ought to be able to come up with an answer with the tools we have. What I'll be talking about is the intense study to study a small number of patients with the disorder here on the NIH campus. So what is the chronic fatigue syndrome? It is when a person develops persistent and clinically impactful fatigue for greater than 6 months. It's coupled with an idea called post-exertional malaise, which is symptoms that come after exertion, such as fatigue, pain, cognitive difficulties, sore throats, lymph node swellings, all after things that should be normally tolerated. There also has to be no alternative explanation for why the person has the symptoms. One thing that's been noted in multiple epidemiological studies is that about 10 to 12% of people who have a significant viral infection or other type of infection develop the chronic fatigue syndrome. And so the focus of the protocol here on campus is post-infectious ME/CFS. To lead this effort, Dr. Avi Nath, the clinical director of NINDS, was tapped to lead the project. And due to my experience with the fibromyalgia community, I was asked to be the lead associate investigator. So the overall hypothesis of the study is that post-infectious myalgic encephalomyelitis and chronic fatigue syndrome is triggered by a viral illness that results in immune-mediated brain dysfunction. 3 phases of the study were planned. but we'll only be focusing today on phase 1, which is a cross-sectional study to deeply phenotype PI-ME/CFS to try to define its pathophysiology. To do this, we separated the study into 2 separate visits. The first visit, which will have groups of 40 that will be participating, is really a deep phenotyping effort. We will be doing a cross-sectional study to do a clinical and biological evaluation of all the participants, and we'll be able to compare our participants with control groups, negative control groups like healthy volunteers, and a positive control group such as people who developed Lyme disease were treated and fully recovered. Patients that make it through this first week with us will be brought back for a second week, which is an exercise stress visit. And the point of that visit is to administer a provocative exercise stress to induce the prime symptom of chronic fatigue syndrome, which is post-exertional malaise. Once we induce that symptom, we will be making serial measurements over 72 hours to get at the understanding of their subjective symptoms, their objective physiology, and to collect biological specimens for further research. We are also adding a couple of extra fun stuff, where we'll be using it to also measure alterations in neurological functioning using transcranial magnetic stimulation and functional magnetic resonance imaging methodologies. So the phenotyping visit is a massive effort. And this slide gives you a sense of all the things that we're doing, the wide scope, and all the different institutes that are involved in pulling this off. Between the clinical assessments done by NINR and NINDS, and symptom profiling done by our nurse practitioner here at NINR, Joy Kreskow, there'll be neuropsychological assessments, occupational therapy assessments, a dietary evaluation, microbiome evaluations using buccal samples as well as stool samples, and that was helped along quite a bit by Wendy Henderson here at NINR. Blood will be collected and studied for immunological alterations, gene expression. We'll be collecting cerebral spinal fluid to do flow cytometry, look at immune markers, do protein discovery, and to look at catecholamines for the first time in the brain. Peripheral blood mononuclear cells, or PBMCs, will be collected to allow for a bunch of novel experiments, including rodent model development and tissue model development. Neuroimaging will be done during the visit 1 using sort of basic anatomic MRI for the brain using gadolinium. A provocative tilt table test will be done to look at autonomic function, and mitochondrial function will be analyzed mostly with the help of Leorey Saligan in his mitochondrial lab. This is sort of a sample of the schedule that we have for our visit 1 phenotyping. This is for a healthy volunteer. For patients, it's double the length. You can see that everything is really tightly scheduled, and we need a crack team to make sure that everything goes according to plan. Those that make it through the first week are invited back for a second week, our exercise stress visit. And this consists of, it centers around a cardiopulmonary exercise test, which is pictured here. Essentially a bicycling test where the participants are pushed to their subjective maximum with our encouragement. And the idea is for them to go as far and as long as they can before they have to give up. It's shown that when you do this to patients with ME/CFS, that they will develop PEM pretty reliably. After that, we will be doing a whole bunch of measurements. NCCIH, one of our sister institutes, actually had a number of focus groups develop a special qualitative interview that we'll be using for this week. NINR has contributed their knowledge of patient-reported outcome questionnaires to match with those interviews. Blood samples, stool samples will be collected, and we will be doing all of this in the evenings in the metabolic chamber here through help with NIDDK, which is a measure of total body energy consumption. When we combine total body energy consumption with measurements of mitochondrial function, we'll have an idea of energy use at the total body level and at the cellular level concomitantly. We'll also be doing EEG measurements of these patients while they are in the metabolic chamber using a portable EEG machine. Lumbar punctures will be measured, there'll be neurocognitive testing, and there's also some novel tests that we developed using functional MRI. We will be inducing muscular fatigue and cognitive fatigue within the scanning environment and looking at changes in blood oxygen level dependent function to get a sense of what is the neurological signal of fatigue. We'll also be using transcranial magnetic stimulation, which allows us to directly stimulate portions of the brain to control the brain's output, in this case in terms of motor function. We will capture their motor function with the transcranial magnetic stimulation and make their muscles fatigue while we measure what happens. And so we will get real novel ideas of what happens to these patients in a variety of ways to create fatigue. This, again, is another very complicated and time-sensitive protocol, and you can see this is sort of a sense of the scheduling, the timing that's required to pull it off. And to do that, we have Team Tired, and a number of them actually showed up today because they're very kind and sweet. We have Angelique Gavin, who's our patient care coordinator in the middle. Anita Jones is our research nurse. Joy Kreskow from NINR is our nurse practitioner. And our two IRTAs, Intramural Research Training awardees, are Benjamin Coleman and Ashley Williams, and Ashley presented our poster today at the poster session. So far, we're doing okay. We started recruiting last October, and since then, we've had 10 healthy volunteers come through. Our 8th patient is on campus this week for the visit 1. And our first patient completed the visit 2 last month. We are currently booked through December, and people seem to think that we're on schedule. With that, I'd like to acknowledge all the different people and different institutes that they hail from that have contributed to the development and implementation of this protocol. It's a wide list that really involves almost every institute here at NIH. Thank you.
So for this portion, what we're going to do is a panel discussion. I'm going to lead off with several questions, and then we're going to open it up to questions from council, so you will have an opportunity to explore this a little bit further. Again, the questions are related to the theme of collaborations. So in general, each of you have demonstrated several successful collaborations here at NIH. As the scientific director, I found that that's the way that we need to function, is these collaborations across ICs in order to leverage all of our resources appropriately. But there are always challenges that come up with collaborations. Can you address some barriers that you have overcome in order to successfully collaborate with other institutes or with others? Leo, or?
Sure, thank you. I think the major challenge that I've faced over time, and I've been pretty persistent in knocking on doors, but really to convey what you can bring to the table, and it's been, and to a number of intramural investigators, there hasn't been a lot of nursing expertise intramurally that has really been able to work with a number of these expertise on campus. So being able to express what your goal, and being able to also share the skill that you can share and contribute to advance the science of both collaborators is very important, and that seems to me the key to for collaborations, at least for my framework to work.
I have to admit, things haven't gone so bad for me. There hasn't been a whole lot of problems of getting things moving forward. I think one of the hardest parts is really understanding the people that you're trying to work with. You can't really give people a top-down definition of what they should be doing for you when you try to collaborate, which is one way to sort of establish things or to create a type of scientific protocols. But you have to sort of appreciate the independence of your collaborators, and the reason why they were drawn to science to begin with is to sort of express themselves and to think through the issues by themselves. To successfully collaborate, you have to appreciate that and help them in their journey as much as they have to help you in yours.
Thank you. Leo, if you could, you spoke a little bit about animal model, collaborating with animal models, and that's not something that NINR specifically had a lot going on before you came and wanted to do that. Can you talk about collaborating along that line?
Thank you. Yeah, that's one of the activities and one of the, I would say, achievements that have really, at least for my program, has pushed my science forward. And talking to my mentors and other experts, I said, it's really an important area for you if you really wanted to investigate mechanistic, if you go deeper into mechanistic investigation, that animal models is really important. Knowing that the resources in NINR and also the ability to gain access to most of these programs or core labs that are available in bigger ICs that oftentimes are close to other ICs was challenging. But chipping away on those challenges can really provide you with these huge opportunities. And once you see, they see your intent, but also the outcome, that especially after you publish your first paper from the animal model that you've collaborated with, the more resources they tend to provide, which is really fascinating. And that really has provided us with this great opportunity to expand deeper. And now with my postdoc here, who's now a shared postdoc with NCI because they really wanted to collaborate with us, is a great opportunity to not only have this great, direct access with NCI resources, but also the expertise that NCI can provide us in improving the rigor of our investigations.
Thank you. Brian, I think most people did catch this, but you are a physician. One of the questions that I would like for you to respond to is, what do you see as strengths and unique contributions that nursing science can bring to the ME, chronic fatigue syndrome, research collaborations at NIH?
Well, nursing has always had the vantage point of dealing with symptoms. And symptoms are really what are the bugaboo for almost all the individual scientists and medical collaborators that I have, whether they're coming from mast cell disorders or endocrine disorders or muscle disorders, rheumatologic disorders, inflammatory disorders. The key of the realm between them all, what holds them all together, are the similarity of the symptoms, or the distressing fatigue and the terrible pain and all the things that they're not able to deal with. In fact, they focus so much on their particular disorder that when we come to them and we talk about symptoms, all they want is our help. Nursing seems to be uniquely situated to sit between all of these people. They seem to be the best situated for such collaborations. For symptoms have always been the bread and butter of nursing practice and science.
Thank you. At this point, I would like to open it up to questions from council members, if there are any. Nancy, go ahead.
Hi, thank you. I have two questions and they're related. One of them is, it's interesting as someone who also does symptom science, is that obviously people from other disciplines, and you nicely answered the question about nursing, we think of symptoms as being very disease-oriented, and yet our NINR research program is looking at symptoms in many ways across conditions. And first of all, for Leo, I was wondering sort of what is the response when you go to other more disease-focused institutes and you talk about your interest in fatigue, right? And not a disease, not prostate cancer isn't the focus, it's fatigue. So I wondered about that. But then the other question I have is a little bit more about the science, and that is, to what extent, so your work, as I understand it, has been largely about prostate cancer, and so to what extent do you think in the future some of your findings might be generalizable or testable in other populations with fatigue?
Perfect question. Thank you. So that's a great conversation that's been going on here intramurally, with especially not only with our collaborators with Brian, but also in general across institutes, and it made it easier moving forward to pitch in the idea of a symptom science center. And because the idea of that is really a disease neutral entity where I bring in, and I explained it to several collaborators, that we really wanted to bring in your patients in because of their symptoms. We want them to be referred to us because of their fatigue. In that way, we will be able to first validate our initial findings from this local prostate cancer population, if it's generalizable across medical conditions, but really understand if there's a shared mechanism for fatigue across this clinical population, as well as any distinct, mostly probably downstream pathways that may be able to distinguish fatigue symptoms for each of these clinical populations. So that is exciting. That phase of developing the center is very exciting, as well as it's easily relatable, and it's easily, you can sell it easy to other collaborators because of that idea.
Any other questions?
I appreciate the good presentation that both of you did around collaboration. I primarily work in an academic setting where we always talk about team science. And one of the things that I kind of tend to think about is that your work is mostly in the laboratory, but some of us do our work in the communities. And I'm trying to see, listening to your presentation, what kind of lessons did you kind of gather from some of this that you could help inform some of us who do a lot of community-based work that we could learn from? And then the other piece is the whole issue of sustainability. How can you sustain some of this over a period of time so that people don't start falling by the wayside and not continuing the collaborative process?
Yeah, so that's the next phase, because as part of the tenure track process, the initial first half of that phase is developing and generating a lot of hypothesis as possible, initial evidence. And my goal, hopefully, for the next part of my tenure track process is really to reach out to extramural community who does a lot of community work to validate our findings, whether this can be validated in the community setting. We've started to work to reach out to different investigators, collaborators for that. And sustainability, again, is really to follow up to make sure that this validation process goes on, but not only unilateral, but across different areas in terms of clinical populations, but also demographic.